This is a topic that is probably my favorite topic of all the things we're working on. It's probably the hardest thing we've ever worked on. And it's going to be, as a talk, a little different from what I think you're probably accustomed to, in the sense that I'm not going to be able to give you an answer. And in fact, it's not even trivial to phrase the questions, but I'm going to be trying to phrase the questions. There are some issues that one can define pretty clearly, but the underlying notion of where life came from, or even what life is just an extremely interesting question. And I want to start with one of the meta issues, which is particularly interesting to me as a chemist about this. And it has to do with the way different subjects are thought about. So if you consider the question of science and big unsolved questions, you can ask, where do chemistry, physics, biology, and so on stand in the scheme of things? And I think if you were to talk to most people about what physics does, they would have a pretty clear answer that it's the origin of everything in dark matter and dark energy and nuclear weapons and quantized space-time and all the rest of it. And don't misunderstand me, I am absolutely for quantized space-time. I just don't have any idea what it means. Then you go to biology, and biology does disease and differentiation and development and what's an animal and things of that kind, and people understand that. And then astronomy is stars. Got it. They blow up. They make fantastic pictures. And you go to chemistry, and it's not completely clear what the answer is. And there's a little bit the feeling that you get when you go to what used to be called the UK. I don't know what we will call it in the future. Where if you hear an announcement on the radio, it will say the sciences and chemistry. And that carries something with you. So what does chemistry do? It is true that it makes better glue. But there are a series of problems, and this is one, where I think that the problems that chemistry must address fall in this category of being very big ones. And I would argue that the origin and nature of life is not a biological question. It's really kinetics and molecules and how they come together to make something that is completely unexpected. I mean, it's really an interesting subject. Then there are others that come after that, such as sentience. And some of the problems that come up in the origin of life come up in the origin of sentience, but it's a little bit more complicated subject. So we're going to talk about it only very briefly, only at the end. Now, let me show you a couple of slides that I showed some of you before, but I want to make a point about this. If you look from left to right in these slides, you understand that this is the natural order of things. If you do the same thing the other way around, it's the unnatural order. And the question is, we all say it's perfectly obvious that you don't demix milk and coffee. But it's worth the effort stopping for a moment and asking why it's obvious that you don't demix milk and coffee. Well, it is not a trivial question. I mean, yes, it has to do with the entropy of mixing, and it has to do with conversion of potential energy into heat and things of that kind. But do stop for just a second and ask about it, because it's very intimately connected to the subject I'm going to talk about, which is the origin of life, which has a little bit this character. So I showed you this before, which is we know how to make a chicken into chicken soup. The question is, can you make chicken soup into chicken? And we don't know how to do that, but it's a little bit even worse than that, because it's really this. So suppose you have something of this kind, which is, I think, what many people would argue is the plausible cradle for life. And then you pour the chicken soup into it, and out of it comes something like that. And it seems very hard to understand how that can happen. How do you go from a system that's disordered, dilute, contains all kinds of other stuff, mostly other stuff, how do you go from that spontaneously, not only to a more ordered state, but also to a state that has the characteristic that, depending upon how you want to phrase it, it's self-evolving or self-assembling. We know how to do that once you get to life, but we don't know how to do it at the very beginning, and that's the reason, at least one reason, why I find the subject so interesting. So the basic issue, we know that molecules are not alive. We know that the cell is molecules. Chemical reactions are not alive. Cells are assemblies of reactions molecules, but cells are alive. So what happened? How does a cell, which is a collection of reacting molecules, become alive? And there are various answers about that. The one that I'm going to come back to is that I think that life is simply a name that we give to a certain kind of reaction, set of reactions. But that makes life, in a sense, a question of networks, and we don't understand how that works. So let me give you a couple of questions to think about during the course of what comes from here. So the first question is, where and when and how did biological life start? And we have a little bit of an idea about that, but it raises an interesting question of, could there be other kinds of life other than the life we know? And the second question is, was the start deterministic or random? And what I mean by that is, in every planet that's a water planet and that has a distribution of elements, is life inevitable? Is it something that will always happen? Or is this just a quirk that happened once per galaxy or once per universe or once per all of recorded time? And I think at the moment we don't know that. You can make guesses about each of these, but I don't think we know the answer. So this has to do with statistics and probabilities, and that has to do with knowing what these reactions are. How does dissipation allow chaos to become order? And we know that you can go from a disordered system to a more ordered system if the free energy of the system goes in the right direction. I mean, this is the second law. But having said that, understanding how you couple dissipation of free energy to the creation of order in detail is not at all a trivial question here. So was it chaos? Or, one of the questions you can ask is, there something else that we're missing in this? Where do alive and not alive separate? And I'll come back to this subject again at the end. But we have two words for something which is not clearly a binary distinction. I mean, I don't know that there's a bright line in between life and not alive, alive and not alive. It may be a continuum. And if it's a continuum, what are the properties in between and what are the properties beyond either end? These are questions that are very, very important when one is thinking about other forms of life. So those are interesting. And there's a question which is relevant to this subject, which is early life and current life. Is current life a reflection of what happened at the beginning, or was it really different at the beginning? Was evolution sufficiently large scale that the traces of what was the first evolving system totally different? And I don't think at the moment we know the answer to that question. And so this is going to prove very relevant to one of the principal topics that I want to talk about in terms of the sociology of this field. And then, is life more than a name for just a set of molecular processes? And there are people who feel passionately on this subject both ways. So is life just like a flame in a sense? And then finally, if one, like me and others, have set off to study this, how do we know when we've been successful? What is the measure that you've gotten over some hump and you're beginning to see something that says that we sort of know what's going on? So where did life start? And we don't actually know where, but let's assume it started on this Earth. And when, we actually have a pretty good idea. The formation of Earth itself, the formation of the planet, was about 4.6 billion years ago. And that's fairly accurately dated. The first, this is what's called the end of the period of heavy bombardment and ocean evaporators, as represented by this. What does that mean? And it means that the things that were falling in this period were big enough that you ask the question, this object comes in, falls through the atmosphere, falls into an ocean. What happens to the ocean? And the answer is it evaporates. So the surface of the planet is then covered with superheated steam for 100,000 years or something like that. And that doesn't sound like a really great place for something as fragile as a cell to come together. The age of the oldest fossilized bacteria, this is still a question of some debate, but it's in this period. So, 4.2, 4.3 to maybe 3.5 giga years ago. And some of the oldest traces are, in fact, in Western Australia. But there are arguments as to what's real and what's a geological thing. This question, how did different sets of molecules and reactions unite? And a point which is actually quite an interesting point, which is that you can argue that life emerged alarmingly early. That is, if this were something which was really a random event, thinking about the difficulty of what has to have happened, it seems that it must have happened pretty rapidly. Now, it's true, there's a lot of ocean, and a billion years here or there is a lot. But the fact that it happened as early as it did, I mean, the planet's surface was barely enough to sustain things without boiling them off. Has raised some people to raise the question that maybe it came from somewhere else, so-called panspermia. And if it did, it doesn't really change the situation because you can then ask the question of where it started there. You just don't know the time scale. Now, in more detail, there's some things here that are plausible but a little surprising to chemists. First is that the planet at that point was largely a water planet. There are people who argue about whether it was completely underwater or mostly underwater. It was probably mostly underwater with some land above water, probably 10% of the above water land that we now have. The continental plates were probably unformed. There's discussion, extensive volcanism, because if there were plates, it was thin relative to what we have now. Lots of geothermal activity. The sun was cool and very UV rich. So there was a lot of stuff going on in the upper atmosphere that produced big gradients in oxidation and things of that sort. There was continuing impaction, which may have been important because it's interesting that when even a pretty small meteor runs into the atmosphere, you ask what happens to the stuff that's in front of the meteor as the meteor comes in at faster than the speed of sound, and the answer is it just piles up. So a meteor simply punches a hole through the atmosphere that's 5 kilometers across, takes all that stuff, ionizes it into a plasma, and then smashes it into whatever it happens to run into. And there's the potential for doing a lot of chemistry there, but we don't know a lot about how that chemistry works. We know something about the atmosphere. There was maybe oxygen in the very upper regions. Probably the upper regions, in fact, were fairly oxidizing. At the surface, it was mostly CO2 and N2 and water. And there are arguments about how much fixed nitrogen and methane and oxygen there were. Probably a lot of methane from geothermal sources, probably not a lot of oxygen. Who knows about this? And then the oceans were acidic, probably pH 5, mildly reducing because they were bright red, mostly iron. And then there was a lot of what I call pond scum. And pond scum means that the atmosphere had been swept off a number of times into outer regions of the solar system and subjected to this UV-rich sun's irradiation. And what you tend to get under those circumstances are molecules like HCN, which have the characteristic that they have a very strong bond in a small number of pieces, because generally, thermodynamically, you favor things entropically under those conditions that have broken up the molecule into the maximum number of pieces. So when you see people doing synthesis in this area, you'll find a lot of reliance on HCN and H2S and acetylene and things of that sort, which seem a little implausible, and on Earth they probably are, but in this period in the formation of the planet, they were probably okay. So I put this picture up again to prejudice you, because I think this is what we're ultimately thinking about. Whether this was on land or underwater, I don't know. But the virtue of having it on land is that it also accumulated the stuff that was falling down. It was subject to UV radiation. It just seems like a good sort of concentrator and reactor for a lot of things. Now we can ask a little bit now about life itself as we know it. And a good question is one that we can answer. What is the simplest cellular form of life right now? And this is probably it. It's an organism known as Mycoplasma genitalium. Herb Ventnor has done a lot of work with this. And you can look at the genome of this. The genome is 5.8 megabases. And there are in the order of 800 proteins expressed. And of those 800 proteins, we know what in the order of somewhere between 550 and 600 of them are. And you can make guesses about some of the others. But it just gives you a sense that this is a bag of molecules that make up this number of proteins which do whatever life does. So that's the introduction. Now we're going to go on and talk about some of these other subjects. And just to give you a little bit of orientation, one of the disagreements in this community, there's a schism right down the center between those who believe in what's called the RNA world, which says that, I don't fall in the RNA world, so I'm going to put this in a slightly prejudiced way. The RNA world says it is so obvious that life must have come from RNA, which was both a catalyst and a memory molecule, that let's just assume that there was RNA and then go on from there. And then the other part, and this is me, says I can't see how to get to RNA, so I don't care whether it was a catalyst and a memory molecule. I don't see how to get there. And there's quite a difference between these 2 points of view. We'll talk about this a bit. We'll talk about some of the results from our work in chemical fossils and about dissipative networks. I'm just going to talk briefly at the end about information in the context of this subject. So, what does this cell do for a living? It replicates itself, and it makes the pieces for doing replication and controls that. And the replication guys are the core of the RNA world, and the metabolism guys worry about where the pieces came from. So the basic schema is that you start with, and we all agree with this basic structure, somewhere there's simple chemicals like H2S and CO2 and HCN, and then a lot of geochemistry. And the geochemistry is intensely interesting and not really very well understood by current chemistry. We don't know a lot about geochemistry, but it's much more complicated than you would think. So simple molecules make more complex molecules, and that's fine. Somewhere there are precursors to RNA. Somehow the precursors to RNA become RNA. RNA becomes DNA, and we sort of know why that happened in terms of stability. So that you can either start intellectually by saying, we know about DNA, we know a lot about RNA, we can ask about that, and let's just assume that this happens somehow because this is all so satisfying and plausible. And the other point of view says, we know about this, but we don't understand how to connect the two. And those are sort of the two different approaches to the problem. Now, what about the RNA world? Why is it so satisfying? Well, RNA is a very interesting molecule. Let's start with DNA. DNA is, of course, the double helix. It's the memory molecule, and we understand a lot about DNA. It contains information in base pairs, but more to the point, the cell puts in a lot of effort to make DNA out of RNA because you get long-term stability that way, long-term chemical stability. But RNA has the characteristic that it looks a lot like DNA in terms of a sequence of bases, which in many circumstances will base pair. And also, RNA is a catalyst. So if it's both a catalyst and in principle, both in principle a catalyst and in principle a memory molecule, it must have been the thing that started off this particular approach. And part of the evidence sort of favoring this is the ribosome, which is, of course, a key molecule in replication of proteins. And it has the characteristic that you can think of it, depending upon your point of view, as a matrix of proteins with RNA plastered in between, or I think more plausibly, as a giant catalyst made out of RNA with proteins stabilizing the whole business. But it's a mixture of these two. It's about half and half. The problems with this point of view is that RNA, we can't figure out how to get there, in my view. Further, RNA is a catalyst, but it's not a very good catalyst. What it mostly does is catalyze reactions that involve phosphates, and it isn't a very good, it looks like it should be a good memory molecule, but it isn't really. So You know, yes, these are good steps along the way, but it's not obvious that that's the final thing. So one of the questions that you can ask there is the plausible question of, okay, so RNA isn't the right thing as we know it now, and either for catalysis or for memory, but maybe we're missing some steps. Maybe there was something like RNA which would work better, but it's disappeared through evolution. And I don't think anyone has a good suggestion as to what that might be, but it certainly is a possibility. Now, the other approach is to say metabolism. And there's been some absolutely brilliant chemistry done by Albert Eschenmoser and Leslie Orgel and John Sutherland and Christian Dedeuve and many others who've thought about the kinds of molecules that you can make in an organic laboratory. And they have done amazing stuff. And particularly, I would point to Eschenmoser and Sutherland. Eschenmoser had a very nice synthesis of vitamin B12 that involved nothing more than HCN. So he made vitamin B12, which is a very complex molecule, starting just from those pieces. The trouble with that was that the synthesis, which was absolutely brilliant, also involved 20 years of very, very skilled synthetic technicians in Zurich in well-defined laboratory conditions. And whether that really tells you something about what could have happened in a wildly disordered planet, I don't know. I happen to think not, but I could be wrong. So this is an issue. You can make very important things, and Sutherland has even found a way of going from simple pieces directly to a base as a precursor for RNA. Whether this applies or not is a question. So the question to arcane conditions, that is conditions in the peribiotic world, is that there's no obvious route to RNA using this kind of thing or to proteins. The laboratory conditions under which this all happened, smokers and volcanoes and lightnings and toxic compounds, don't bear much resemblance to what's going on up here. And then there are a series of things having to do particularly with concentration. Because if you're going to have two molecules react with one another at a finite rate, they have to have some proximity to one another. And if they're dilute, present but dilute in an ocean, what is the probability that you will get reactions that lead to the kind of thing that you want with the cell? Probably not high. There are hints that I think are interesting, and one is relevant to what I'm going to tell you later, that there might be other ways of doing it. So, for example, you can make peptides, you can make proteins, little proteins, without having a ribosome. And the method that's probably most important now is something which is actually pretty complicated in its own right, but it involves a piece of machinery in which the sequence of the protein is determined by the sequence of subunits, protein subunits, which instead of using RNA and activated amino acids, use thioesters. And this is one reason why Christian Dedeuve talked about the thioester world. This particular thing makes molecules of this sort and a bunch of other bacterially derived antibiotics that are oligopeptides, but probably more important, exactly the same kind of chemistry makes fatty acids, which are the lipids that make up the cell membrane. So it's a very successful reaction pathway. Now, just a couple of points here. And this is as a reminder of something you already know. When we talk about equilibrium systems in chemistry, we sort of understand what we're doing. We have pretty well established rules for doing equilibrium. We do understand the sources of free energy that could be used in origin of life. So sunlight and radioactive decay, heat in the center of the Earth coming out in thermal gradients, concentration gradients, things of that sort. It turns out that although there are many gradients in, for example, temperature between a kilometer down in the Earth and the surface, we don't have very good methods of harnessing that kind of energy, free energy, to do chemistry with. And there is a point, and this is for those of you who are students primarily to remember, when we talk about free energy, we write delta G0 equals delta H0 minus T delta S0, and we phrase this in kilocalories per mole as if it were a fundamental property. And actually, it's the wrong way of writing the equation. You should really write it as delta G0 over T equals delta H0 over T minus delta S0, which is a statement of second law of thermodynamics. So everything is basically the second law. And there's no separate thing which is free energy. It's just entropy that runs everything. The problem with this is that, of course, this is all in equilibrium conditions. And exactly how to think about something that might be a thermodynamics for a dissipative system, we do not know, even though some very smart people have thought about the problem. So, in a sense, this issue begins to boil down to the nature of dynamic and dissipative networks, and we don't have a very good understanding of these, either theoretically or empirically. And there are some important issues in autoamplification and autocatalysis that I'll come to, and there's the whole field of what we call systems chemistry and biochemistry, which is a subject of active discussion, but it is not correct to say that the field has solved its internal problems. So then let me turn to some experimental results. And we've taken the following kind of approach. One is that we have been interested in looking at what I call Archean compatible chemistry. That is, chemistry that we have chosen in the hope that it might actually be directly relevant to the conditions of the peribiotic Earth as opposed to the conditions of a modern synthetic laboratory. It's a different kind of chemistry. And I'll give you a couple of examples just to give you a notion in ATP and the origin of the potassium-sodium gradient that characterizes all of life right now, and then things like isoprenoids and metallic cofactors. I'm going to sketch and only sketch the area in which we've spent the most work. I've talked about this briefly here before, but it's the issue of a thioester-based network and what one can get out of that in terms of self-organizing behavior. And then finally a bit on information. So, molecular fossils. The argument here is purely hypothetical, and it may be wrong, but the argument is the following, that if you're thinking about the origin of life, what you might want to do to trace back to whatever was going on then is to look at features that are common to all life now. Now, maybe it evolved later, but if it was a good idea at the very beginning, maybe it stuck. And what are some examples? Well, all life has the characteristic that it's high on potassium on the inside, and it lives in an environment in which sodium is rich on the outside. So, in fact, the storage of free energy in terms of the potassium-sodium ratio is one of the great commonalities across life. We look at this now and we say, well, that's obvious. If you've got potassium and sodium, you can make a storage battery by simply concentrating the dilute thing. The problem with that is that, of course, before there was life, there was no sentience, and there was certainly no thermodynamic analysis. So something else has to have happened then. Polyphosphates, that means ATP. One can make a pretty clear case where that kind of come from. Thioesters, I'm going to talk about that. They're common throughout life. ATP and the common cofactors are common throughout life. Where did these things come from? And then these major metabolic cycles, like the Krebs cycle and glycolysis, and the light-sensitive molecules that are used almost everywhere to harvest sunlight, so porphyrins and retinol and flavins and things of that kind. So let's start with ATP because that one is easy. This is, of course, the activating chemistry, the equivalent of the chloride, the carbonyl chloride in all of synthetic chemistry that's used to activate oxygen. And all one has to do is to take phosphate in the presence of urea on a hot rock, and you heat it at 100 degrees, and what you see by capillary electrophoresis is this series of peaks, these sort of jagged peaks there, each of which is another step longer polymer of polyphosphate. So that it's not hard to take phosphate and condense it into polymers, and in fact there are organisms even now that use polyphosphate as a principal energy storage source, and so it's natural to say that triphosphate attached to some recognition handle could have been the origin of ATP. That seems okay. The sodium-potassium gradient. This is more complicated, but the question here is there a way in which you could plausibly have gotten to media that were high in potassium and low in sodium, even though the environment is very rich in sodium and poor in potassium? And to do this, you go to geochemistry. And without going through the details, very common rocks are basalts and feldspars and zeolites of various sorts and clays. These are all basically aluminosilicates of various kinds. And if you take one of these mixtures, you take a mixture of potassium sulfate and sodium sulfate, and you put in a solution with something that becomes, well, you start with a molecular sieve and a mixture of sodium and potassium, or alumina, a mixture of sodium and potassium, allow these to convert into their stable form. The crystals that are formed here, the mineral that's formed here, is 100% potassium, essentially, and with alumina, it's 100% potassium. So that you can find geochemical processes which naturally crystallize things in terms of very high potassium forms. So the idea would be that you start with a sodium-potassium mixture of some kind, you do a crystallization on the surface of a volcano, and then you wash away the sodium that's left, and what you're left with is a potassium-rich brine. And that would be the place where, in this argument, you would have started something. And then, although we haven't done isoprenoids, I think you can make a good story there. And the story suggests this idea of missing things. The way isoprenoids are made in current isoprenoids, for those of you who are not organic chemists, are things that are derived from isoprene formally, which is this C5 molecule. And the way it's made now is a pretty complicated process by which you start with acetyl-CoA, which is a thioester. You make this rather complicated molecule by some processes that we understand and end up with this, and it's all under tight control. But it turns out that the most stable and most abundant olefin in C4 that you can make is isobutylene, very stable molecule. And formaldehyde very plausibly could have been formed by reaction of methane produced geochemically with oxygen produced by photolysis of water in the upper atmosphere. So there was almost certainly formaldehyde there. And a touch of acid makes this, or precursors of this, and then we know that acid from that begins to form the bonds that form this. So here I think what happened was a natural process that went this way that's evolved over time into this. And that touches on the RNA issue in saying that there's no life that does this now, it's been lost, but the products have been preserved, and it seems plausible that one could have started with RNA precursors, something else that then evolved into RNA in the same general way. So one can work through a number of the important catalysts that are present in everywhere. Urease is the enzyme that hydrolyzes urea. Carbonic anhydrase is a key enzyme that catalyzes the equilibration of carbon dioxide and CO2, carbon dioxide and carbonate. The ferredoxins are really important in various ways in redox chemistry, nitrogenase, whatever. These all have active sites that look just like minerals. So the idea that they may have formed by just taking colloids in smokers and wrapping stabilizing proteins or something around them makes pretty good sense. So I think one can find plausible ways of making a very large number of cofactors, cofactor-containing enzymes from pieces that are either naturally occurring colloidal minerals or simple molecules that could have been formed by the kind of processes that we're familiar with. Now, can you put all this together? And here I'm giving you a kind of community view that probably the most plausible environment to think of for these earliest life forms was this, or maybe it was a deep smoker, or maybe it was a dying sea. These have the characteristic that they concentrate because you have dilute solutions of things coming in. they evaporate water in the heat, and so you get concentration, you get UV irradiation, you get colloidal catalytic minerals being produced. You can imagine ways in which you get a high potassium brine. There are a lot of pieces that come together into a story that is not a convincing story for anything, but it's a start. There is, however, a little glitch, or at least in my view, there's a glitch. And you see it in, for example, metabolic processes that are common to all life, basically, such as the Krebs cycle. The Krebs cycle, if you look at it, it has a couple of thioesters. That's good. We're comfortable with thioesters, although we don't know exactly how to make them. There are things that are iron-sulfur clusters. Those are colloids from hot springs. These sulfur compounds were certainly not acetyl-CoA, but they can have come from hot springs. This can have come from evaporation. There are other steps. Here's another colloid. You know, the pieces all look together okay, but the problem is this one, that each one you can make an explanation for where it kind of come from, but we don't have any idea of how it was all integrated into a piece. High potassium brine, isobutylene plus acid, a deep smoker, probably hydrocarbon junk in deep smokers. How does this all get together? No idea. I mean, not a clue right now. So, we're moving, having not declared success in point A, moving on to the problem that I think is really the problem. This is the era of the RNA world, the field of the RNA world. We're interested in this problem, which is networks, and are there alternatives to RNA in terms of memory? I mean, can you have a memory in a cell that isn't based on RNA or DNA? And we've taken an approach to this, which is, we'll see. It's to say that the cell is a dissipative system. It takes fuel, which is glucose and other nutrients, and oxygen, and it burns them in a reactor. It makes CO2 and other forms of waste, and what it does with that is to generate the components that make the cell. And we're going to model that in terms of something which seems ridiculously far away, which is a flame. And a flame takes a reactant, which is a methane rather than glucose, and it takes oxygen, and it burns them, and it makes heat and light and CO2 and water. So how do you put all that together? How do we learn something from this that is in any sense relevant to this? And we're going to do it by using chemical engineering to model the cell with what those of you who are chemical engineers will recognize as a continuous stir tank reactor, except that a cell doesn't require stirring because it's so small that diffusive mixing is as fast as you need it to be. Now we're going to do one other thing here, which is to try to deal with the problem of complex behavior. And if you remember, a cell has the characteristic that it grows and then it replicates, and it grows and it replicates. So there's a periodic behavior to it. Where else do you see periodic behavior of that sort? And one of the places that I've always been curious about is that those of you who have seen and even worked with gas stoves that have these ring burners will notice that when you turn down the flame, as the flame goes down, you'll often get a rotating circular behavior. I mean, very interesting. Where does that come from? And what's happening there is that as the flame goes down, locally it will use up all the methane, but it will illuminate the flamelet next to it, and it goes around in a circle, and by the time flame comes back to here, this will have gotten enough methane in it that it can go off again. That's the sort of general process. You're working just on the edge of instability. So what we're going to do is to work with a flame in which, in essence, a flame in which we have a chemical reaction, not a methane reaction, but it's one in which we are going to try to get this kind of cyclic behavior. And the system that we're going to work with is thioesters. And I'm going to tell you the result for those of you who are organic chemists that are interesting and important. The reason for working with this system is that thioesters have a remarkable characteristic, which is this carbon-sulfur bond can exchange with other carbon-sulfur bonds essentially quantitatively rather than hydrolyzing, which makes it an ideal system for working with, an ideal reactant for working with systems. So this is another argument for the thioester kind of world. And ignoring the details, the relative rates of hydrolysis of the system we studied in detail, the relative rates of exchange reactions, one thiol for another, versus hydrolysis is in the order of 100,000 to 1, which is enough to do anything we need to do. So it's a very, very good reaction. And I think a number of us have the intuition that the only reactions that have the robustness to contribute to the development of complexity are reactions that go in essentially quantitative yield. So here's the reaction. Don't bother to look at it because it's too complicated to understand. I just want to put it up there to make the case, the following case, that if I look at this as a long-term organic chemist, I can't make any sense out of it. It's too many things going on. And I can write the differential equations and, in fact, solve them, or simulate them, but that doesn't help me very much either. So it's a problem that comes up fairly often in the chemistry of complex systems, that you can get things to work empirically, but by doing so, you don't necessarily learn everything you want to learn. So understanding and doing are not the same thing. And what we've begun to appreciate is that there are other languages for thinking about these kinds of complex systems that make them much easier to work with. And the one that we're pursuing at this point is control theory, which is a language and approach to the simulation and control of complex systems such as, for example, a petroleum refinery. And we've broken this problem up into pieces that actually can be understood. And the entire system I'm going to show you, all of this stuff, consists of 3 steps. There's a step that's an auto-amplifying step in which once a set of reactions reaches a certain critical value, a reaction goes off exponentially. And for many complex behaviors, you need auto-amplification. To set this off, we need a trigger, and the trigger is going to be composed of a process that makes the triggering molecule competing with another process that destroys the triggering molecule. And there's a 3rd step, which is washing out of the reactor, which enables you to control the concentrations of many of the pieces all at the same time. Those are the components. And when you embody this in an actual device, it's pretty straightforward, a little complicated to do, but syringes that feed the reactants into a mixer, the mixer goes into a stirrer, you measure products that come out, and there's a lot of technical detail in this, but the basic structure is very easy to understand. And when you do that, this is the behavior you see. That is, if you look, for example, at the products, you will see as you increase the rate at which you feed the reactants into the reactor, a steady state, you change that rate, you get to another steady state, you change that, you get to another steady state, you change, you get to another one, goes down, and then you reverse that process here to here to here, and you don't go back this way, you go a different way. So the system shows what's called bistability, that is, proceeding in one direction, you get this series of behaviors, and proceeding in the reverse of this behavior, you get a totally different set of reactions. So bistability, you see oscillations, so the reactions show spontaneous oscillations like the division of cells, and may be completely unrelated, but it's that kind of stability. And you see something that I'll come back to, which is that as you change the control parameter here, which is the space velocity, which is basically the rate at which you feed reactants into the reactor, you see this complex behavior over a range, but when you make it faster or slower, that behavior disappears. So it's a behavior that's defined over in a limited set of circumstances, and as many processes with life. If you freeze me solid, I don't work. If you heat me to 100 degrees, I don't work. So I don't think it's worth the effort here to go through the details other than just to show a couple of essential features. In the autoamplification reaction, what one does is to start with a reaction that has a thiol group. You do chemistry, which was designed, and at the end of a single cycle of reaction, you get 2 thiols. It is amazingly difficult in chemistry to design such processes. There are very few of them. An explosion is one, crystallization arguably is one, maybe electrolysis deposition, but for reasons that I don't understand, you just don't see very many auto-amplifying or autocatalytic reactions. And I think it's probably an area where there's great room for discovery. The trigger is simply to take one of these things and to hydrolyze it. It's the thiol that starts the reaction, and we can control how much of that's here by having a reaction that destroys that. And you can see, this is in red, the concentration of this inhibitor. As it's consumed, the auto-amplifying reaction does nothing so long as it's there. As soon as it goes away, the reaction goes off autocatalytically. So that's the triggering process. And then inhibition is a related kind of thing. It's even more complicated to understand. But this behavior here, this is the first time that anyone has taken a system which is biologically relevant in the sense of containing kinds of chemistry that could have occurred and demonstrated a complex, bistable, oscillating behavior of the sort that you see here. And it's quite interesting, and this has got a number of people very excited. Now, one thing that fits into this that is a broadly interesting subject to me is the issue of robustness. What you find in these systems is that they do this oscillation, but they do it only a very narrow range. Interestingly, though, you take related molecules which do not oscillate, you put them in, and they're entrained by the oscillating reaction, so the whole system oscillates. And so what you can do is to make a system that oscillates, but over a much broader range of conditions by including a mixture of things which do what they do. It raises a question which has to do with Darwin. And we have a picture of Darwinian evolution which says that what life does is to optimize its behavior to make the fastest cheetah or the best beak for a goldfinch or whatever it might be. An alternative view is to say that all of that is a view of life which is life as it is now in a highly optimized form. And at the beginning, what in fact we may have needed was the most robust reaction as possible, which is actually a quite different set of processes. And so these kinds of things provide a very good way of studying that kind of thing. Now let me finish by just talking briefly about this question of memory. Because one of the characteristics of life is that it has a memory. That's DNA. And the characteristic of DNA is it's carefully tuned so that in each cycle of replication there's a certain amount of error. Those errors generate the mutations which allow the system to evolve. So mutations and error are a big deal. So when we think about memory and think about evolution, we have to think about these kinds of questions. And I raise these for you to consider as we go on here. How do we know whether something is alive or dead? I mean, what's the criterion we're going to use? If it's alive, it can mutate and evolve. If it's dead, presumably it can't. But is there a distinction that we can make between live and dead or not alive? And we're talking about something of this sort. Well, I'll come back to this in just a moment. But is a frozen embryo alive? It does no chemistry. It doesn't evolve. It doesn't do anything. But you warm it up, and it becomes palpably alive. Is there a sharp point where something happens? What about a virus, or what about a city? Is a city alive? It certainly can evolve. It has a memory. It can divide. It can do all the other things that, in principle, a cell can do, but it's certainly not a cell. And then an interesting question that we're beginning to think about is, if we have a hard time understanding how something becomes alive, can we understand how things die? And that's not so easy either, because we think of death in a pretty straightforward way as the brain stops working. But that doesn't have all that much to do, arguably, with whether something, an organism, is alive or dead, because many organisms don't have live brains anyway, living or dead. So I want to show you just a couple of plots. Along this axis, I'm going to have aliveness, and I don't know what that is. And along this axis, I'm going to have complicatedness or complexity, which are somewhat different. And I don't know exactly what that is either. But pictures of this would be, in one sense, a binary. That is, something is dead up to a certain point, or it's not alive, where a switch flips and it becomes alive. That's binary. Okay. Another would be something of this sort. It would be sort of exponential, or maybe it would be sigmoidal. If it's exponential, it becomes more complicated, it becomes more alive, and you could then ask where we are in this. Or you could say, and I think this is the kind of in the back of the mind interpretation that we tend to have, which is that life started with nothing, it got more and more complicated until it reached the ultimate crown of creation, which is us. And maybe that's right, or maybe it's not right. But then there are other things of this sort, and the one that I think keeps cropping up is folded bifurcations. And in a folded bifurcation, the governing equation that would be an exponential here actually looks like this. And it has the characteristic that things are going along, and they're getting more complicated, and then at a certain point, something happens, and it doesn't continue up in that direction. It undergoes a transition. It hops. But then when you turn down the complexity, it comes down here. It doesn't reverse the path. It does this. It's bistable in that sense. And I showed you an example of that with the thiol chemistry. But could this be what we should be imagining? Or should we be imagining this? Or should we be imagining this? I mean, what is the proper description of how something goes from not alive to alive? And I don't think we have, this is not a question of origin of life, it's a broader question. I don't think we have a good answer to that. So when you're thinking about that, I think most people who are scientists are now sufficiently marinated in the world of binary arithmetic that we tend to have a binary prejudice. And I want to just do a quick comparison of binary, as in integrated circuits, with whatever it is that goes on with enzymes. So when one thinks about an integrated circuit, you're thinking about a collection of switches that are mostly on or off. There's a common working fluid, which is electrons. Everything works with electrons, and everything works at a common voltage between the rail and ground. The function of this system is defined by the fixed topography of the system. That is, what's connected to what. The switches are binary, and they're really fast. Memory is capacitors or magnetic domains that are switched on or off. That's the system is, in a sense, non-Boltzmann. That is, the on and off are equally energetic. That's not quite true, but we don't need to worry about it. But the whole system is organized by a common clock, though all the things go through steps in synchrony. And that's a way of thinking about it. Biochemistry is fundamentally different. It's not this, it's this. Everything is sigmoidal. The cell is a sac, so the dimensions are set by diffusion. There's no topographical organization. There are multiple working fluids. Every compound in principle can influence every one of the other switches. It doesn't. Slightly synchronized, maybe. You get sigmoidal responses. It's very slow, and it has memory embedded in DNA or RNA, or maybe in networks. So they're fundamentally different systems. And should we think about them the same way? How do we think about control? And I don't want to go through any of this other than to point out that in biology, you use control strategies that look like this. For example, for high amplification, molecule A will go to B, and B will be converted back to A, but B will be a catalyst for C goes to D, and D will be a catalyst for E goes to F. And if you look at an important reaction like forming a blood clot, it looks like that. Why does it have that structure? And I don't think we have any idea why it has that structure. But it's a very interesting problem. So I've raised with you a number of questions, and I don't think that I can answer really any of them, which is why it's such an interesting area. But I think this final slide is one that's good to leave the subject with. You can ask the question, what is life? And perhaps as with an electron, and I mentioned this in the previous slide, when I talk to my friends who are physicists and say, what is an electron? What they say is, you can't ask that question. You can't ask the question of what an electron is. You can only ask the question of what it does. And so it may be that we have a slightly confused impression with this subject of asking what it is as opposed to asking what it does. And you come out with quite different answers when you think about two different things that way. So it is a wonderful subject to think about. It actually is one of the big problems in science. It is something which I think only chemistry can do, or chemistry and chemical engineering. And it's something for us to have a good deal of fun working with. I can't at the moment think of a single application of this, but that's okay. Thank you very much for the invitation.
Thank you, George. The floor's open for questions if we have some from the audience.
So when, for example, people at NASA, when they're looking for another life form outside of the Earth, why are they always looking for water? Is water really necessary for another life form? Could there be another life form that can use something else after in place of the water?
The answer to that is we don't know. And if you ask what could another solvent be, you can think of other solvents. Well, let's say one of the characteristics of water is that you can put a hydrocarbon shell around water in terms of a lipid and make a compartment. And that's probably necessary for life as it develops. There are other solvents in which you could do that, for example, HCN. The problem is that it's not so easy to make oceans of HCN, and pure HCN has the characteristic that it's a neat liquid, but it blows up. So HCN, I think, would have a tendency to go wrong. My colleague Frank Westheimer, ex-colleague Frank Westheimer, wrote a paper at one point which he called Why Phosphate? And he simply went through all of biochemistry as he knew it and asked if there was anything that he could think of that could replace phosphate in water-containing life. And he came up with the conclusion that nothing could replace phosphate. So that doesn't mean you couldn't think of something else, but it does mean it's going to be a hard thing to do. So my guess is that if you want to look for other forms, other planets with life, it's good to look for water. And the figure, which I think I've told some of you, you hold your finger at that size roughly, at about an arm's length, and point it in any direction, and you see a billion galaxies, that angle, some tens of billion galaxies. And each of those galaxies is 10 billion stars. And most of those stars, well, many of those stars will have planets, and at least some of those planets are bound to be water. So I think there's effectively an infinite number of water planets around there. How long life lasts is a different question. It may be that we're a billion-year phenomenon, but we don't know.
There's a question over here. Sorry.
The first development was on alkaline hydrothermal vents as being a frame for the one to emerge, and there seems to be a very plausible environment for complex hydrogen.
Yep. Yeah, the answer, that's again, it's a neat question. We don't know very many alkaline vents. There are a lot more acidic vents. That doesn't mean that a slightly idiosyncratic reaction couldn't have happened in alkaline ones. And I agree that there's a lot of good chemistry that you can do in alkaline systems that you can't do in acidic systems. On the other hand, the whole ocean was acidic, so that if you're doing something in a vent and it's then spewing out into the environment, you'd have to do it in alkaline conditions and have it stable under acidic conditions. Yeah. Yeah. I agree. I think they're very interesting. And for those of you who don't follow this subject, there is an argument that the initial life, if you want to put it that way, the initial cell was not a lipid container, but it was a crack in the rock. And the crack was lined with catalytically active minerals, and it had stuff flowing by it. It may have had a plug of hydrocarbon junk at the end, but it didn't look at all like a cell as we now know it. It looked like a geological feature. And it developed later on, and it could have certainly developed in an alkaline environment.
So that was really nice. My question is about the RNA sort of if you thought that some of the work you showed here today could still lead to the development of RNA species, and then from there we can see where we're going to DNA.
Yep. Absolutely, yes. The question is there reaction chemistry that goes from the simple pieces that we can imagine to RNA, or did RNA come by through something that looked like RNA but was simpler and easier to come by, or did it just happen that, you know, some alkaline environment, whatever, coughed up RNA? I don't see how you cough up RNA. But John Sutherland thinks that it's possible, and in specially controlled conditions, he has made one base.
You've raised the difficulties with answering the what is life question by focusing on things, entities, and saying, is this alive, is that alive? Maybe we can overcome part of the difficulty by thinking of life not as a series of entities, but as a process. That process converted a simple cell, let's say simple life, into complex life, giraffes, etc. And maybe that same process, exactly that same process, but formulated in chemical terms, converted inanimate matter, let's say dead matter, into simplest life. And therefore, by thinking about life as a process and focusing less on specific chemicals or materials, one can get a better understanding of what life is.
I think that's exactly right, in fact. And as I said, to me, life is like a flame. That is, it is a collection of reactions which has a certain set of properties. And so the reason we focus on reactions and entities is to, in a sense, provide a plausibility to things. How could you have gotten complexity? But a flame, even a methane-oxygen flame, is probably 10,000 reactions. And we're very happy with a flame without ever knowing how a flame actually operates. And my guess, if I had to guess, I would say life is going to be like that. So it is a series of processes and networks which have a property in their aggregate, but maybe have different forms and detail in between. What's so interesting about life as a process, you take methane, oxygen, you heat it, and all of the free energy, potential free energy there, is dumped as heat. What happens in life is it is exquisitely good at taking that overall free energy gap and pulling off little pieces of it and using each little piece in a different way. And the question is really, how would you go from something that's a big gradient, undifferentiated, into a series of little ones that build constructively to something that becomes more complicated? And I don't understand how to do that right now.
Hi, George. I'm an Archean geologist, and I'm going to come to dinner with you, and I'm interested to talk a bit more. And so excuse this question, I'm not a chemist, but about 3 slides before the end, you showed a series of reactions that sort of stepped one to the other. And I guess intuitively, that looks like a very nice approach. And prior to that, you talked about the difficulty of some chemical reactions going in binary and duplicating themselves. But that stepwise reaction about developing complexity in a piecemeal way where each reaction is not at all dependent on what follows on, but it comes to a certain stage and then it gets added on by complexity. So that intuitively looks very exciting to me. Is that a common way that chemistry works?
It's a common way that chemistry works. I mean, if you think about complex synthesis, you do reactions and reactions and reactions, and each one makes a more complicated piece, and at the end, you have a very complicated structure. However, each is separate. It's run in isolation, and the conditions are typically incompatible from one to another. And so what seems to happen here, maybe this also occurred in a series of different thermal ponds or something of that kind. It's just that we don't know. So the question of how you break up something into these pieces that make stuff that fits together into a network that shows complex behavior is an interesting question. And that system that I showed you with the network is probably not directly relevant to anything involving life, except that the chemistry is the kind of chemistry that you could easily imagine occurring in an alkaline vent or even in an acidic vent. So it is not wildly out of whack with what might have happened. So it's a small step.
If I can just follow up with a short teaser, I think it's very interesting, this question about alkalinity versus acidity.
Right.
One of the things I think that's underappreciated about hot spring fields is that within a single field, which may have tens to hundreds of pools, you get the entire range from acid to alkaline, and they can exchange information. You talked before about the oceans being problematic in terms of them being very diluted, having a problem with concentration. So I think that's something to consider about the differences between an oceanic versus a freshwater geothermal system.
One of the things which I've come to appreciate in this is that as somebody who's been studying chemistry for a long while, there's an entire universe of stuff having to do with geochemistry that is critically involved in this story, since this is undoubtedly where much of the free energy came from, and the catalysts and the reactants and all the rest of that. I don't know anything about it at all, nothing. And it's a glaring hole in this subject right now. We just don't know the chemistry. So you and I are going to have a nice dinner together.
Yeah. Let's thank George very much for a second.